Hi, this is Les and Amy from the RevGenetics podcast. You can listen to the podcast on YouTube and now on Spotify, where we do a deep dive on supplements and longevity studies.
So today we are taking a look inside your refrigerator.
But not for a midnight snack this time.
No, not this time. We're looking at the hidden heroes, the tiny workforce that turns milk into yogurt.
And cabbage into sauerkraut.
Exactly. But here's the twist. We're looking at a brand new 2026 review, Precision Biomanufacturing with Lactic Acid Bacteria, which shows how scientists are giving these ancient bacteria a massive high-tech upgrade.
It's a huge shift, isn't it? We're going from just using them for flavor
to treating them like programmable microscopic factories. I mean, the whole idea is wild.
So we're talking about reprogramming the same things that make cheese to, what,
Right.
Produce medicine?
Medicine, high-value vitamins, even personalized superfoods.
It's kind of like finding out your grandmother's old wooden spoon has a USB port and can, run algorithms.
That's actually a really, really good way to put it. We're seeing ancient food science meet cutting-edge biology.
Okay, so before we get to the sci-fi part where my yogurt is, curing diabetes,
Which we will get to.
we have to ground this. The paper talks about lactic acid bacteria or lab. What are they exactly?
Well, they're a group of bacteria that have been around humans for thousands of years. You find them on plants, in dairy,
And in our gut, right?
and crucially in our gut. They are what's called generally recognized as safe, or GRAS.
So the good guys.
Exactly. The friendly neighbors of the bacterial world. Their main job historically has been fermentation.
And in simple terms, what's that?
Think of them as tiny chefs. They eat carbohydrates, so sugars like lactose in milk,
Okay.
and their body converts that sugar into energy. But the byproduct of that meal is lactic acid.
They eat sugar and boop acid.
Essentially, yes.
Yeah.
And that acid is the key. It lowers the pH of the food, making it acidic.
And bad bacteria don't like acid.
The nasty ones, like Salmonella or E. coli, just can't survive in that environment. The LAB creates a sort of biological force field.
So they sour the food to save it. It's biological warfare, but we get a tasty snack out of it.
Precisely. And for centuries, that was the whole deal.
Hmm.
But this paper is about the shift from that relationship to what they call precision biomanufacturing.
Okay, break that down for me. Old-fashioned cheese making versus this precision method.
Okay, so traditional fermentation is like gardening in a wild forest. You throw some seeds out there, you know what will probably grow, but you're really at the mercy of the environment.
It's artisanal, which is a nice word for unpredictable.
Right. Now, precision biomanufacturing is like a high-tech, sealed hydroponic greenhouse.
Where you control everything.
Everything. The light, the humidity, the nutrient drip, the genetics of every single seed. We are designing the bacteria to do a specific job and controlling the environment to make them as efficient as possible.
So we're not just letting nature run its course. We're giving it a timesheet and a performance review.
In a way. And to do that, the paper highlights a toolkit that's just completely changed biology. The first big one is CRISPR-Cas9.
Right, everyone's heard of CRISPR. Usually with things like designer babies and sci-fi movies. What's it doing in my food?
It's editing the recipe. I mean, bacteria run on code, right? Their DNA. Right. CRISPR is basically a molecular search and replace function. It finds a specific sequence in the DNA,
And
the Cas9 protein acts like a pair of scissors. It just snip. It cuts it.
So you can cut a line of code that says, make the milk sour.
You can cut it, or more importantly, you can paste in something new. You can insert a gene that says, hey, instead of just making acid, I want you to make vitamin B12.
Wow. So you're rewriting their whole purpose?
We're diverting their energy from just surviving to manufacturing things we want.
That sounds incredibly hard to get right. You can't just hope you pasted the gene in the right place.
No, and that is where the second tool comes in. Artificial intelligence and what they call digital twins.
Digital twins. I've heard that for, jet engines. You build a digital version to test it.
It is the exact same concept, but for biology. A digital twin here is a hyper-realistic computer simulation of the fermentation tank. It models everything. The bacteria's metabolism, the temperature.
So before you waste a bunch of milk, you run the simulation.
Thousands of times.
Mm-hmm.
The AI plays out all the scenarios. What if we raise the temp by half a degree? What if we add more sugar at hour 4?
It's like a flight simulator for bacteria. The AI crashes the plane 1,000,000 times so the real pilot doesn't.
Exactly. Just cuts down on all that expensive trial and error. But to build a good simulation, you need really good data.
And how do you get that? This leads to that other term, right? Multi-omics. Sounds complicated.
Sounds it, but it's pretty logical. Omics just means studying the whole system. So you have genomics.
The blueprint, the
Yep.
DNA instructions.
Right. Then there's transcriptomics, which is seeing which of those instructions are actually being read at any given moment.
So genomics is the whole library. Transcriptomics is the book you have open on your desk.
Perfect analogy. And then there's proteomics, which studies the proteins they're actually building, and metabolomics, which looks at the chemicals they're producing.
It's like reading the bacteria's diary. You know exactly what makes them productive.
And once you know that, you can feed it back into the digital twin, refine the CRISPR edits, and create this super efficient biofactory. It's a complete feedback loop.
That's impressive.
It really is.
I want to take a brief second here to give a shout out to RevGenetics for letting us dig into this topic.
Absolutely. RevGenetics has always been about, the rigorous science behind natural compounds.
And it really fits, doesn't it? This study is all about using hard data to optimize what nature gave us. In the same way, RevGenetics focuses on verifying the science behind things like NMN and other longevity supplements.
Exactly. It's not about guessing. It's about understanding the mechanism. And that's what separates the real science from the noise.
Okay, so let's get to the so what part. We've built these super smart bacteria. What are they making? Because if all this is just for sharper cheddar, I'm going to be a little let down.
It is definitely not just for cheese.
Yeah.
The applications in medicine are, well, they're revolutionary. The review highlights a major breakthrough with GLP-1.
GLP-1, that's everywhere right now. It's the thing behind Ozempic and Wegovy for diabetes and weight loss.
Correct. GLP-1 is a peptide hormone that tells your brain you're full. But the drugs are mostly injections.
Because if you just ate it in a pill, your stomach acid would destroy it.
Your stomach thinks it's a steak. Exactly. But here's the breakthrough. Scientists have engineered a strain of bacteria to actually produce GLP-1.
So the bacteria is the factory.
It's the factory and the delivery vehicle. You eat the yogurt with these bacteria. They survive the stomach acid, set up shop in your intestines.
And start making the GLP-1 right there.
Right there, directly at the site where it's needed.
That's incredible. So instead of a weekly shot, you could have a prescription yogurt.
That is the vision. It turns a medical treatment into a dietary habit. I mean, compliance would be so much better. No needles, just a spoon.
And you'd get a smoother dose, probably, not the spiking trash you might get from an injection.
That is the theory. A steady state release. They're calling it a living therapeutic.
Living therapeutic, I like that. But they're not stopping there, are they? The paper mentioned nutraceuticals.
Yeah, foods that act like drugs. One of the big ones they're making is NMN nicotinamide mononucleotide.
A huge topic in the longevity space for boosting NAD plus rank.
Right. And making NMN chemically is complex and expensive, but these bacteria can be tweaked to just overproduce it during fermentation.
So you could have a drink that's not just probiotic, but is actively loaded with anti-aging molecules made right in the bottle.
Exactly.
Yeah.
And they're doing the same for things like GLC and AC, which is great for joint health, and GABA.
GABA, that's the relaxation chemical in your brain, right? The chill out one.
Yes. And normally it's hard for GABA supplements to get to the brain, but producing it in the gut might be way more effective. Imagine a stress relief sauerkraut.
I would buy that by the case. But, okay, devil's advocate here. High-tech factories usually have a cost. Is this sustainable?
That's actually one of the strongest arguments for this. The paper calls it the circular bioeconomy.
So recycling.
Basically. These bacteria need to eat, but they aren't picky. They can be designed to eat agricultural waste.
Like what?
Like whey from cheese making.
Huh?
For every pound of cheese, you get like 9 pounds of whey. It's a huge waste product.
So you take the trash from the cheese factory.
You feed it to your engineered bacteria, and they turn that waste into, say, high-value B vitamins or NMN.
That's amazing, turning trash into treasure, literally.
They're also looking at fruit peels, cocoa shells.
Oh.
It's incredibly efficient. It decouples making valuable stuff from using up scarce resources.
Okay, so we have waste-eating, drug-producing bacteria, but the paper mentioned something even more futuristic. Smart neuronutrition.
This is personalization. Your gut microbiome is totally unique, like a fingerprint.
So a one-size-fits-all yogurt doesn't really make sense.
Right. The goal here is to use AI to analyze your specific microbiome. You send in a sample, and the AI sees what you're missing.
No.
Then it designs a fermented food just for you. It picks the exact bacterial strains that will produce the compounds your body needs.
So if I'm an athlete, it could design a mix to help with muscle recovery. If I'm studying, a brain-boosting blend.
Precisely. It's food that's functionally designed to optimize your personal biology.
That is wild.
Hmm.
But I also saw the paper mention postbiotics. That's different, right?
Yes. Very important distinction. Probiotics are live bacteria. Postbiotics are inanimate. They're dead.
Why would I want to eat dead bacteria?
It sounds weird, but it's brilliant for safety and manufacturing. Sometimes you don't need the bacteria to be alive. Just parts of their cell walls can trigger a positive immune response.
So you get the benefit without any risk of the bacteria, overgrowing or mutating.
Exactly. And you don't have to worry about keeping them alive on a grocery store shelf. They're shelf stable. It solves a huge logistical problem.
Zombie bacteria might be the safer bet.
Let's stick with postbiotics, but yeah.
Before we wrap up, I loved that the paper took a moment to acknowledge the history here.
Yes, paying tribute to the pioneers. They specifically highlighted female pioneers like Rosalind Franklin.
The woman behind Photo 51.
Exactly. Her work was crucial to figuring out the double helix structure of DNA. Without knowing the shape of the latter, we could never have learned to edit it with CRISPR.
And Jennifer Doudna, of course.
A Nobel laureate for her work on the CRISPR system itself. We are truly standing on the shoulders of giants.
It's just a beautiful arc. From the first person who accidentally discovered yogurt to someone like Rosalind Franklin seeing the code of life.
To an AI today designing a bacteria that could treat a chronic disease. It totally redefines our relationship with food. It's not just fuel anymore. It's becoming information.
We hope today's podcast on published science gave you something useful to take away.
Thank you for listening to the RevGenetics podcast.
RevGenetics.com for the best longevity supplements on Earth.